All right, here's our experiment, the Yeast Contemplation Project by Ashley and Elma. And first thing you want to do is we have to set up our plates, our yeast sample for the weeks to come. And you will need an agar plate and the Bunsen burner in your sterile loop. And of course, wear your proper PEE. And to collect your bacteria sample, you should have unknowns and your alphas from your instructor. And collecting the unknown first is what we ended up doing, the unknown number 3. And we hovered that sterile loop over the Bunsen burner, collected the unknown 3 sample, smeared and streaked it onto our new agar plate in the indicated area, and then reheated that over the Bunsen burner and repeated the process with other bacteria samples. And here in this picture, you can see Elma with her very proper safety gear on, and our agar plate, after basically 2 weeks, I think it ended up being, or a week and a half, there's something along those lines, some extra time to incubate. And you can really see in the picture on the far right, our unknown sample is kind of all over the place, and our alpha isolated and mixed with our unknown. It's hard to tell, but there is some peach light red coloring. You can see it a little bit better in the middle picture, although it almost kind of looks yellow with the agar color, but if you looked closer in the natural light. But as I was saying there at the end, that you can see it a lot better in the natural light to tell it does have some color. And now the next thing we're going to be talking about is ADE mutations, and there is a lot of information on that slide, so just bear with me on that. But an ADE mutation is a mutation involving adenine, and in this case it is a base in DNA and RNA, and specifically in yeast mutations, the ADE stands for adenine oxytrophy. This means that the strain of yeast will not survive unless adenine is added into the system as the growth medium. And because of that addition of adenine, you get that red color, but we will talk about that fancy chemical interaction in a later slide and in a later assignment. And I left some examples of those ADE mutations off to the side, and those were from our lecture PowerPoint, just to give us and you an idea. And on this next slide, I'm talking about A and alpha mating types. And here we're talking about those mating types, and A, or MAT, little a, in genes are for the haploids, and this determines 2 opposite mating types. And it compares the existing strands that you are using in your sample with tester strands to solidify those results. And I did include a little infographic about kind of what it does, because I am still unfamiliar with it, but we're learning. And also at the top, those are 2 yeast molecules actually coming together. And I also included the diploid. It's just a little explanation about when 2 of the haploids come together and create the diploid offspring. They cannot mate with other diploids just because of the unique structure with the A and the Z that is given from the A, or MAT-A genes. And I found that information from the Kansas State Yeast Genetics. And this is the last one, effects on yeast morphology. And there could be multiple changes in the yeast structure just because of those levels of adenine, according to the FEMS Yeast Research Volume 14. This came out in 2014, coincidentally. And this kind of showed you, I included their infographic. I found it actually really interesting, even though we are not to that level. But they use a different yeast sample than us, obviously. But it kind of just showed me that the levels changed, resulting in no more production, halted production, and whatnot because of that level of adenine.